So today we are privileged to have very distinguished speakers with us from the pharma industry. Sarah Neuhaus from Prevail Therapeutics, which is a wholly owned subsidiary of Eli Lilly and Company, and Dr. Andrew Whitaker from Freeline, who will be speaking to us about their respective clinical trials for Gaucher disease. So to introduce the speakers, Sarah is the medical director of two ongoing clinical trials for gene therapy programs at Prevail. So the first one is called Proceed, which is a clinical trial for adult patients with type 1 Gaucher disease. And the second one is Provide, which is a clinical study for pediatric patients with type 2 Gaucher disease. Sarah received her medical degree from the Philadelphia College of Osteopathic Medicine. She completed her child neurology training at Duke's University Medical Center, followed by a combined clinical and research fellowship in neuromuscular neurology and neurogenetics at the Johns Hopkins Hospital and the NIH. Meanwhile, our second speaker, Dr. Andrews, after qualifying from the Leicester Medical School in 2000, he completed his postgraduate general medical training in the University Hospitals of Leicester NHS Trust, becoming a member of the Royal College of Physicians in 2003. So Dr. Andrew brings a wealth of expertise and experience in cardiovascular research. Dr. Andrews joined AstraZeneca in April 2016, where he worked on multiple translational science and drug development projects across a broad range of areas, and was also a member of the Cardiovascular Safety Knowledge Group. So in January this year, Dr. Andrews joined Freeline Therapeutics as the Senior Medical Director and is excited to now be involved in the development of gene therapy for lysosomal storage disorders. So once again, I would like to thank both our speakers, Sarah and Andrew, for supporting our cause and for contributing to this educational webinar. So before we proceed with with their presentations, I encourage all the attendees to please use the Q&A box to ask any questions they may have. And we will be taking up all the questions after we are done with both the presentations. Also, a quick reminder that we cannot address any questions that contain any sensitive information or patient-specific details. So with that, Andrew, could we please begin with your presentation first?
Yes, thank you very much, Suyog. I'll just share my screen. Hopefully this will work. Can you see my screen okay?
Yes, we can.
Great. Well, thank you very much to the IGA for this opportunity to speak today on behalf of Freeline, and thank you very much to all the people who have joined the webinar to listen to us. And as Suyog says, please put any questions in the Q&A box. So I'm going to talk to you today about our clinical trial, which is a phase 1 trial in patients with Gaucher disease type 1. And we call our trial Galileo 1 for short. And the treatment we're talking about that we're investigating is called FLT 201. Just as a brief overview, this is just an educational webinar, and that all our gene therapies are still investigational treatments, and this is not supposed to be viewed as being as a healthcare advice in any way. So as I'm sure all of you probably know even better than I, that Gaucher disease type 1 is a rare genetic disorder caused by genetic alterations. So abnormalities in a gene which sits within DNA. That means that the body and cells within the body cannot make enough of an enzyme called GCase for short, or glucocerebrosidase. And this enzyme normally works inside cells to prevent buildup of a harmful fatty material. And in patients with Gaucher disease who don't produce enough of this enzyme, the harmful fatty material can build up inside these cells and disrupt their function and cause them to swell. So in the image below, you can see that on the left-hand side, in a healthy function, you have sufficient amount of enzyme produced which breaks down the harmful material. But in Gaucher disease, the reduced or deficiency in the enzyme leads to a buildup of this material inside the cells. And what that means is that throughout the body, where these Gaucher cells can build up, you get impairment or reduction in the function of some of the tissues, and also some of the organs within the body can swell up. And so we find that patients can develop enlargement of their liver and their spleen. The bones can become brittle and painful and at high risk of fracture. And the center of the bone, called the bone marrow, where blood cells are produced, does not work normally. And so there is a tendency towards a low blood count or anemia, and a reduction in certain parts of the blood that are involved in clotting, which can lead to an increased risk of bleeding. And less commonly, some patients may be at risk of developing problems with lungs as well. The current treatments, which many of you will be well aware of, is either enzyme replacement therapy, where patients attend to have an infusion of enzyme every 2 weeks, continually throughout their life to replace the missing enzyme. And this means that you get very high levels of the enzyme around the time of the infusion, but then the levels drop away and become quite low to very low between the infusions. Alternatively, there is a tablet form of treatment called substrate reduction therapy that people take each day, and that reduces how much harmful material is made in the first place. What we're hoping to achieve with gene therapy, both ourselves and Prevail and other people interested in this field, is that the hope is that by delivering a working version of the gene that can make the missing enzyme, if we can deliver that with a single therapy, and then the body is able to use that gene to produce the enzyme continually and release that into the bloodstream, which circulates around the body, and then all the cells in the body can take up that enzyme, and that can then clear the harmful material from the cells. The way we do this is, Prevail, I'm sure will tell you about theirs, but it's very similar, I suspect, is that we use a virus that does not cause disease, but is able to get into the human body and is able to enter human cells. And this type of virus is called an AAV, or an adeno-associated virus. And these kind of viruses are out there in the environment all the time, and many people are exposed to them. And they don't cause infectious disease in humans, but they are able to get into the body. And so we utilize that as a way to deliver our gene therapy into the human body. So we take one of these types of genes and we modify it so that we take away some of the viral particles or the viral DNA, and we change the virus so that it's no longer able to replicate. So the amount of virus that we put into the body with the gene therapy cannot become increased. And then we use the empty sort of viral particle as a carriage capsule to deliver the new gene that we want to give for the therapy. So we put the new gene inside that, and then we can infuse that virus, which contains the new gene, that gets into the cells within the body. And in our case, we use a virus that is specific to targeting human liver cells, to go to the liver to deliver the gene, and then we use the human liver cells to make the new enzyme from that new gene. So in summary, in Gaucher disease, the gene that people have that causes Gaucher disease is not working properly, and that means you don't have enough enzyme and you get a buildup of the harmful substance. When we put in the new treatment or the gene therapy, which is called FLT201, we deliver that through an infusion into a bloodstream via an arm vein, for example. The virus particles that contain the gene therapy go to the liver cells and are taken inside the liver cells, and then the liver cells use that copy of the gene to make the new enzyme. The liver cells then release the new enzyme into the bloodstream. It travels around the bloodstream, around the whole body, and the other cells in the body can then soak up that new enzyme to clear the substrate or the harmful substance from the other cells. Now, our gene therapy, similar to Prevail's, does not enter your own DNA. It does not modify your own DNA that you were born with. It produces a new separate section of DNA that sits inside a cell, but is separate to your natural DNA, and just separately works there as a template for your cells to be able to make the new enzyme. So we're not changing any of your genes as such, or taking any out, or modifying your DNA. We're just introducing a new small section of DNA specifically for this enzyme. Our therapy FLT201 has been slightly modified so that the enzyme that we produce, the GCase enzyme we produce, lasts longer in the bloodstream and within the cells, so it doesn't get broken down by the body as fast as natural GCase, and this means that it can be around for longer to build up in the cells and break down the harmful material. Here is a diagram of what our ongoing clinical trial looks like. So this is the Galileo 1. It's a phase 1 trial, which is a first in human. So phase 1 is the first time that a drug gets tested in humans. And this is a very typical design of what's called a dose escalation safety study. So we start at a low dose and we see how patients who receive this dose react and how well it works. And then depending on the information, we decide whether we increase the dose, for example, to this mid dose and potentially even up to the higher doses. We don't have to go up to all of these doses, and we review the information that we get at each dose after each patient to see how well it's working and how safe it is. And as well as the company, there is also what's called a data monitoring board, which is made up of experts who are not part of the company, who look at that information and determine whether the gene therapy is safe and is doing what it should be doing. There is what's known as a stagger between patients. So when we dose the first patient, we wait at least 4 weeks to see how things are going for that patient before making a decision to dose the second patient, for example. And then after that patient, it's at least another 4 weeks. So there is a gap between patients so that you are able to take the time to evaluate the safety. And safety is the primary objective of this trial, but of course we are interested in seeing how well it works in making the enzyme and clearing the harmful material. So far, we've dosed 3 patients in this study, all in the low dose cohort, so the bottom dose cohort here, and all 3 are doing well. Just as a note, it is common in gene therapy trials that as part of the treatment, patients also receive some medication to suppress or dampen down the immune system to reduce any inflammation that can sometimes happen in the body, particularly occasionally in the liver. And so we have a regime where we start using a steroid therapy at week 3 after we've given the gene therapy. And that continues for around 3 months or so. And sometimes we give a second medication in addition to the steroids to help dampen down the immune system. As I mentioned, we've dosed 3 patients so far in our study. And these 3 people are now, further on than this, but the first patient is around 4 to 5 months after the dose. The second patient is around 3 to 4 months after the dose, and the first patient is around 6 weeks. The third patient, sorry, is around 6 weeks after the dose. From a safety perspective, all the infusions have been well tolerated. During the infusions, the patients felt well and didn't have any side effects. There have been no serious side effects. We've seen no evidence of liver inflammation, and we can detect this by looking at blood tests that tell us whether the liver cells are inflamed or irritated. We haven't seen any of those in the patients. There has been 2 mild side effects which have thought to be possibly related to the gene therapy. They happened in the first patient about a week after dosing, and both got better by themselves. And there have been some mild to moderate side effects which are not serious, related to the steroid medicines that we use to suppress the immune system. These were expected based on the known side effects of these types of medication. So far, the safety of this is looking good. And what do we see with regard to how well the gene therapy is working? Well, before entering the trial, we know that all 3 patients had very, very little or very low levels of natural enzyme being produced by their body. And over the course of a few weeks, we saw a gradual increase in enzyme being produced by the gene therapy from the patient's liver and released into the bloodstream. And we can measure this by taking a blood test, and we look at how much enzyme there is in the blood, and then also within the cells within the blood. And we see that over the space of weeks, we see quite quickly that the levels of GCase enzyme in the body become well within the normal level. And is this working or not? It's quite early to be able to say for certain yet. As I mentioned, the first patient is around 4 to 5 months out and has stopped their background therapy. So we're now starting to see whether this is working instead of their previous therapy. And it takes several weeks to a few months to determine whether the gene therapy will be working to have an effect on the harmful material. But in this first patient, who is now, as I mentioned, 4 to 5 months after therapy, we are starting to see a reduction in the substrate levels in this, or the harmful material levels in this patient have begun to reduce from around 3 months onwards. So, as I mentioned, within 1 week after we give the gene therapy, we can detect levels of enzyme in the bloodstream well above normal levels, and they stay above that level. By 4 weeks, we can see that the blood cells, so the white blood cells in the blood, are also starting to show normal levels of GCase enzyme. That tells us that those white blood cells are taking the enzyme up from the bloodstream and taking it inside the cell to work on breaking down the harmful material. And we see that after a month after gene therapy. And as I mentioned, we're starting to see some early signs of reduction in harmful material. As the trial is ongoing, it is open now in 7 countries, 6-7 countries, in around 15 sites. These are generally, this is Brazil, Israel, Germany, Spain, the United Kingdom, and the United States. And this is a trial for patients with Gaucher disease type 1, who are adults, so 18 years or older, and they're already being treated with either enzyme replacement therapy or substrate reduction therapy. A brief, I realize we're at 16 minutes, so a brief summary of what's involved in the trial. So there is a screening period which can take up to 4 months, where your investigator or your physician will determine whether you're suitable for the trial, and you will read the information to determine whether this is something you would like to do or not. And for those patients who are suitable, who wish to take part, then they receive the treatment. And for the first week, it's reasonably busy. People are seen on a daily basis for the first 3 days, then again at the first week. And then patients are followed up for around 9.5 months to see how well it's working and for safety. And then after that, patients enter a long-term follow-up study to be monitored for up to 5 years. This is a quick summary of what's done. So we do regular blood tests very frequently, at least weekly for the first few weeks, and more often to monitor the liver tests. And then things such as scans to monitor changes in your liver and spleen size and see how your lungs are working and how your bones are less often, but happen in the order of between every 4 or 12 weeks. And we do have a clinical trial website if people are interested, which is for patients and their physicians, and further information can be found either by looking at this or contacting our clinical trials website. Thank you. I'll stop sharing my screen now.
So thank you, Dr. Andrews. That was a very informative presentation about the Galileo trial. So I would now like to ask Saga to kindly begin with her presentation.
All right, can I just confirm you can see the proper screen slide?
Yeah,
right.
We can.
Fantastic. Thank you so much. So my name is Sarah Newhouse. I'm the medical director for 2 ongoing Gaucher gene therapy trials at Prevail Therapeutics, which is a wholly owned subsidiary of Eli Lilly. I'm thrilled to talk to you this afternoon and evening. Thank you for joining, and thank you for following the lovely presentation. So we'll be able to really build on each other and talk about 2 exciting trials for Gaucher disease type 1. Just want to give a brief background, same medical disclaimer that the trials we're talking about today are not approved therapies and are still under evaluation for safety and efficacy, or how well they work. So this should not be interpreted as medical advice. Let me give a brief outline. I'll introduce you to our company, our group, Prevail, share a little what's going on in terms of our clinical trials, talk a little bit in depth about gene therapy in general, because there's a lot of discussion in the news, social media, TV, documentaries about gene therapy, how it works, what it is, and all the excitement around it. So I think it's important to know the types of trials that we're discussing today. Then we'll talk about Prevail's approach, which was mentioned as an AAV, an adeno-associated virus approach, and then get into more details around the PROCEED trial, which is actually designed for patients with Gaucher type 1, but we specify peripheral manifestations of Gaucher disease, and we'll talk about that in more detail. So Prevail is focused on leveraging genetic medicine and gene therapies for rare neurodegenerative diseases. And so currently right now, we actually have 3 clinical trials for different types of GBA1 mutations. Many of you know that this same gene, this misspelling in this DNA, can cause multiple types of diseases. So in the earliest stages would be Gaucher disease type 2, most severe in infants, Gaucher disease type 1, which we'll be talking about today, and then can also be associated with a rare form of Parkinson's disease. So we have clinical trials in all 3 of those GBA1 conditions, along with a clinical trial in a completely different area called frontotemporal dementia. So we'll focus on our trial today, but just to give you a view at large of the trials we have ongoing. So before we get into the specifics of the PROCEED gene therapy trial, let's talk about what is gene therapy and what we're discussing today. I'll be building on a lovely explanation already, just to hear it again in a slightly different way with some different cartoons and graphics. The goal of gene therapy is to effectively replace or transfer a healthy copy of a gene into your cells, not replacing, but sort of adding or transferring to your existing DNA. So when we talk about this, I like to make an analogy to a tire, changing out a tire when you have a flat tire on a car. Is if you've ever had this happen, you realize you have to replace the tire that isn't working with another tire, hopefully in your car. You put the broken one in your trunk and you put the functioning tire on your car and hopefully get to your destination. So in this method, we are transferring or adding, some people call this gene addition, to your current existing DNA in your cells, a properly spelled GBA1, which makes the GCase enzyme, but we're not editing, we're not removing, we're not changing anything. So just in this car analogy, you still have the tire in your trunk, you just have that functioning wheel to get you where you need to go. And I think that's important to understand how this specific type of gene therapy is working when there are many things in the news about gene editing and CRISPR and many other technologies that is not what we're doing. That is not this technology. I also want to make a mention that our trials are new and that these trials we're discussing today are first in human, meaning they've gone through animal testing rigorously, usually in different animal models, and are first in human. But gene therapy as a concept, and also AAV gene therapy, has been studied for many, many decades, since the 1980s and early 1990s, so that this concept, although new in this area, has been studied for a very long time. And so we're building on that knowledge and learning every day, every year, helpful information on how to do this successfully. So let me run through the most common gene therapy approaches that are out there and what you may hear, and then talk about what we're doing. So we did just touch briefly on gene therapy versus gene editing. And as we mentioned, gene therapy, also called gene transfer or gene replacement, is providing that new copy of DNA to make an appropriate protein. Whereas editing has to do with going into your DNA that you're born with and changing something. The next sort of circle over to the right is something called in vivo versus ex vivo. And I pause on this briefly because I know that this community is well aware of a previous trial that, although not ongoing, the AVROBIO trial involved a very different method of administering gene therapy. And so I just want to highlight how this is different here. Some gene therapies, such as the two we're discussing today, are delivered directly into a peripheral vein. You get an IV infusion of a virus with the DNA. In other trials and previous approaches that you may have heard, it involves sort of removing cells from your body, editing them in a lab, and retransplanting them, which is a very, very different method. So just to mention, the ones we're discussing today are an infusion and don't involve that perhaps more lengthy process of that removing cells from your body and using a different type of virus. Which leads us to the next circle on the right, which is what types of viruses or vectors or capsids do we use, these sort of delivery agents? We use the natural property of a virus, which has evolved to learn how to get into your body's cells. We use that for good by replacing its viral genes with something healthy to replace a misspelling. And in both of our cases, we use the top, this adeno-associated virus. Adenovirus is typically a common cold, and I say that this is sort of a surfer virus that hangs out with the AAV, with the adenovirus. AAV alone doesn't cause you to get ill. Versus other trials which have used lentivirus, which is different. It can be integrating and it can cause different side effects. Additionally, areas that were different are our approaches to immunosuppression. Some trials use oral medications, as we've both discussed today. We can talk about that more. Typical things such as steroids or other medications that are used to dampen down, reduce the immune response. Versus in some of these ex vivo approaches use perhaps more harsh, more difficult conditioning regimens with chemotherapy. That is not what we use in our study. So just want to really highlight the differences for these trials. And as you're thinking about potentially participating, what would you experience and feel as a patient going through this? What would it be like? So we are doing the following 4 top areas that we do. Therapy, not editing. It's in vivo infused directly. We use AAV and we use prophylactic medication, so that these types of approaches involve typically very short hospital stays and no chemotherapy. All right. So as we discussed as well, and a slightly different graphic, that we utilize this AAV. In our case, we use an AAV9, and that's effectively working as what I like to call a delivery truck, some type of post office truck or an Amazon truck that is shuttling this DNA into your nucleus. And as you can see with the graphic, it actually ends up forming it sort of cohabitates. It lives separately from the DNA that you were born with. And functions to make this properly formed GCase. And I pause here again to say that people have asked, this is not something that if you have a child in the future, you would pass through your DNA to the next generation. Many times people say, if I'm treated, if I have a child in the future, will they be protected against Gaucher disease? So unfortunately, this therapy, maybe for good, for bad, it is a one-time therapy for the patient receiving it. It is not designed to be inherited by the next generation. So because the DNA stays separately in the nucleus and doesn't integrate and doesn't edit yours, it is not passed on through the next generation. So I think that is important to distinguish as well. That reduces some risks. And so that is the stage of this type of AAV-based gene therapy. All right, so with that in hand, let's move to our PROCEED trial. And I do want to mention here that we are accepting patients with peripheral manifestations of Gaucher. Largely, this is patients diagnosed with type 1 Gaucher disease, but we do know that the GBA1 mutations and the Gaucher disease, the way it looks in patients is a spectrum. It's a huge range. And so from type 1 to 2 to 3, people can have different levels of liver, spleen, bone marrow involvement, blood counts, and sometimes neurological symptoms. And so in certain rare cases, and I encourage you to speak with your physician and speak with us directly, there are certain type 3 patients that are mild that could be considered for treatment in the trial. So I do want to distinguish that important difference. So would be something we would need to discuss on a case-by-case basis, but that we are considering patients with peripheral manifestations of the disease, which largely means your organs and blood and bone marrow. At a high level, what does the PROCEED study look like? And what are the goals? As discussed, any first-in-human trial, the goal is safety. How safe is this? Are we able to do this in a way that is not causing harm to patients? How is the therapy tolerated by people who receive it? And so that is the first and primary goal, is safety. Similarly to what was discussed with our colleagues at Freeline, it is also a dose escalation, dose finding study. So is it safe, and can we find the right dose? As mandated by health authorities around the world, all of our regulatory overseers, this trial has to be 5 years long. Usually the first year is the most intense, the first 6 months, but we have to follow all of our patients for 5 years to assess safety appropriately. Our studies are pretty small, looking for a total of around 15 patients, and we're looking for adults aged 18 to 65. We also have around 8 international study sites. Not all of them are open. We can talk about that in detail as well. And as we mentioned, the goal is that this is a one-time therapy. So we're using an AAV9 vector. It is an infusion that is single, it's non-recurring, and that it's delivered by IV using prophylactic immunosuppression. So let's walk through some key inclusion criteria. People are often wondering, which trial should I select and which trial am I eligible for? For the inclusion features, we already discussed the age. We need a clinical and genetic diagnosis of Gaucher. That's usually already something you have had for several years. One of the key features is that you have been on a standard of care therapy for at least 2 years. So for our study, that would be ERT or SRT for a minimum of 2. It could be any of those, mix and match. We know that for many reasons, side effects, travel, patients may change their current therapy for periods of time. So it doesn't have to be the same therapy for 2 years, but that you've been on some approved therapy for that period of time and are stable on your therapy for 3 months prior to the dosing period. In this initial trial, because it's first in human, we do need to test, in this case, patients who are not having the best response to their current therapy. And so what does that mean? That looks like patients who are on ERT or SRT, and despite having tried that therapy or maybe others for 2 years, there are still some symptoms that are not well addressed and are bothering you. So that could be persistent enlarged spleen or a persistently enlarged liver. And those are things that we typically test with an MRI, and we can calculate very specifically the size. Despite therapy, there are still low platelet counts. We call that thrombocytopenia, so that no matter what treatment you've tried, perhaps you have a persistently low platelet count. And then there are several bone symptoms that we include, certain levels of bone pain and crisis, and certain findings on bone imaging. So those would be critical inclusion criteria for our trial. What are some of the exclusion criteria? Who may not be able to participate? So let's talk about the first one. Many questions about what does it mean to have AAV antibodies? What does it mean to be antibody positive or negative? Why does this matter? Why is every gene therapy trial testing this? As we talk about that capsid or shuttle that is taking that good replacement DNA into the cell, into the nucleus, your body doesn't know that this is a good virus, that we have equipped this virus with something to help you. Your body recognizes this, the little envelope, the sort of shuttle, they recognize that as a virus. And so in an immune response, as your body does with every virus, with COVID, with flu, your body creates antibodies to protect against it. And so in this rare case where we want your body to accept this virus, if it has created a certain level of antibodies, it would effectively sort of chew up or block the therapy from being effective. So that's why many of the trials, especially those that are delivered into a vein, into your arm, into a peripheral vein somewhere, have this exclusion criteria. We can't accept patients that have high levels of antibodies against the virus because we worry that the therapy may not be effective. And so that's why that's an important exclusion criteria to pause on. I'll also just mention different trials use different viruses, different little delivery trucks, if you will. If you think about DHL or UPS or the post office, there's different delivery mechanisms. And so if you are not able to participate in one trial, you may be able to participate in another one. So I encourage you to think about that. And also, we don't know how long the antibodies last. And so if you have a high level of antibody, you may be able to test again in a few weeks, a few months. We're not sure how long those stay in your body. So it's something to consider. I think this is an important area of learning for doctors and scientists and companies as we're developing gene therapy. The antibodies are something we think a lot about, both for inclusion and for efficacy of our treatment, and for making sure that we are able to treat and include as many patients as would benefit from our therapies. So apologies for that very long explanation on that inclusion, but it covers a lot of territory that is really important for you to think about and understand when you're considering clinical trials. The others are a bit more straightforward. We use certain immunosuppressants. If you have very bad adverse reactions to those, it wouldn't be safe to participate. If you have significant neurological impairment, and we can discuss this on a case-by-case basis, if there are areas of severe seizures, for example, and history of a total splenectomy would be excluded as well. And then we've had several patients ask about a partial splenectomy. That would be included. We would be able to accept you, but patients that have had their entire spleen removed, because we're using that as a measurement, if your spleen has been removed, we're not able to measure it appropriately. If there are progressive bone diseases that will require surgery, we would not want the trial to interfere with anything that is planned for your medical care. And if there's additional evidence of severe liver and spleen complications. And that's largely to protect from any side effects that may occur in this first-in-human trial, selecting patients that would tolerate well and not have any bad side effects. Typically, these inclusion and exclusion criteria are more narrow, they're tighter in the first study. We have to be very strict. And as we learn more and treat more patients, these are things that we hope can be relaxed and expanded and open to different ages, different types of patients. But for those first trials, you can tell we include a bit of a narrow window to find the right patient to ensure safety and ensure a response for the condition. All right, so our study design looks very, very similar, just slightly different graphics. But as you can tell, we also have 3 dosing levels, a low, middle, and high dose. We have a pre-screening and eligibility time where we check your antibodies, we check the genetics. Some patients are also diagnosed only on enzyme level and may not have genetic testing. If you don't, that is no problem. We're able to provide that as part of the study. I just want to mention patients who may have only been dosed on clinical symptoms or enzyme level. That's something we're able to do. And you enter a dose finding cohort. In this case, just to mention, we have to do that in a sequential manner. We cannot go right to middle or high. We've had patients say, well, can I just start right away in the middle or high dose? We have to do 3 patients at low and then examine if it's safe, move to middle and move to high. We can't start right in the middle or high dose. And this is designed to assess the safety and the response, how much enzyme is being made, how much of that toxic substrate is being reduced. Because the truth is we may not need to go to the middle or high dose. We may have a surprising response at a low dose. And so we don't want to give you more of something that you don't need, and we have to assess that sequentially. And so very similarly, we have a stagger between patients as well. We also assess with an external medical review board, a safety committee that assesses it after each patient, and then makes the decision to move to a middle or high dose if necessary. So we have around 3 patients at each. When a dose level is selected, then we move to the expansion cohort and treat 6 additional patients at that time. We have really important blood and imaging biomarkers that we get every 3 months, the first year, 3, 6, 9, and 12, along with blood biomarkers that we're monitoring as well. And then the visits and the blood work relax a bit as we follow you every 6 months in the later time of the trial. I did want to mention as well, we're trailing right behind that we were able to dose 1 patient in 2023, and I don't have any data, unfortunately, to share today, but look forward to bringing that to the community as soon as I can. So we are also excited to have our first patient dosed. A brief overview of the study schedule and visits. I think this is important to understand. I think many patients have asked us about the travel burden, the visits, how frequent are they, how long do I need to stay, do I have to stay in the hospital? As you can tell, I would say the first month is the most intense in terms of visits, where there are frequent physical exams to check for safety, along with blood safety biomarkers and biomarkers for the Gaucher disease. After month 3, things tend to be a little bit more relaxed in the every 3 to 6 month visits, and after month 12, they're every 6 months. So with these sort of little cartoons, you can see, obviously your doctor will do a physical exam every time and check on you. And then we have those imaging symbols to get an MRI of both your liver and your spleen. We do an abdominal, a belly MRI to check your organ size, as well as an MRI of the bone to check the bone marrow and look for those Gaucher cells and see any improvement. We also do questionnaires to see how you're feeling, how you're functioning, how is your energy, fatigue, bone pain. We want to know how you're feeling. That's really important. This has to both be safe, it has to show us the right blood levels, then you also need to be feeling well and improving. And so this is just a brief review of the type of tests you would do at each study. Just want to mention here, the first month is the most intense, but does not require a hospital stay. So you do not need to sleep overnight in a hospital. We prefer that you're close to the doctor that dosed you for the first month to make sure things are safe, but you don't need to typically spend a night in the hospital. Usually it's, if at all, it's usually the first day for the infusion and a monitoring period. That will depend on the doctor, and that is really the only inpatient, in-hospital stay that is required. So just to wrap up here, I know we probably have a lot of questions to get to, but our study is designed first and foremost safety and efficacy, as we mentioned. We also want to look at very important blood disease biomarkers, which is that GCase enzyme and the GLUCIF, the glucosal sphingosine, also known as lyso-Gb1, that toxic buildup of the fatty substance. We want to look at your bone marrow function, your blood measurements, mainly hemoglobin and platelet count. We want to look at your liver and spleen volume and size volumetrically, And then we have questionnaires to see quality of life, energy improvement, fatigue, and also looking very closely at bone and lung disease progression. These are areas that are typically not well managed by ERT alone. Those are parts of the body that ERT has trouble getting to. And so the questions we want to answer with gene therapy is, can this type of therapy fill in those gaps to be more effective? So let me move quickly. That's a summary we just discussed. Please discuss with your physician. We have our clinical trials number down here. And I do want to mention for this group, we have 4 open sites. Unfortunately, they might be quite far away from our audience today. We're working on opening additional sites, but currently we have 2 sites open in the United States on the East Coast in both Virginia and North Carolina. And we also have 2 sites open in Spain as listed here. But feel free to take a screenshot of this for contact information, reach out to Prevail as well. Travel is covered for patients who are interested. I want to mention that as well in terms of proactive travel reimbursement. I'm sorry, not reimbursement, proactive travel coverage is possible. Patients should not feel financial stress to participate in a clinical trial. We know that the participation alone is burdensome, the travel time is burdensome. So we work very hard together to make sure that those things are covered and that participating is despite the burden and the time, that participation can be as easy as possible for those that are generous to consider participation in our trial. So with that, I'll move to questions, and thank you so much for listening today.
All right. So thank you very much, Andrew and Sarah, for these incredibly informative and enlightening presentations. So I'm sure they have enriched our understanding of these ongoing clinical trials for Gaucher disease. So now we have our floor open for any questions from the audience. And I would just like to remind the audience to just input their questions in the Q&A box, and we'll have them answered one by one. So we'll try to take as many questions as possible, but in case there are any unanswered questions, then we'll ensure these are answered after the webinar is over. So I don't see any questions as of now. So meanwhile, we had a few questions which were sent to us beforehand. So I would just like to go through them one by one, and if maybe both of you could answer them one by one, that would be great. So the first question is, so is gene therapy suitable for all patients? So maybe we could start with Andrew.
Hi, yeah. So the short answer is not every gene therapy is suitable for every patient. So there are certain patients who may have existing medical conditions that may not make them suitable for a gene therapy right now, because as Sarah mentioned, in the first in human clinical trials, we're very careful as to what kind of patients should go into those, and it tends to be patients who have the least other medical problems. And then the second part is there are often restrictions regarding age. So for example, both our trials are in adults right now, so 18 years and older. We don't have an upper age limit. I believe, Sarah, was yours 65 or 70? Sarah will know, 65, I think.
65, 18 to 65. It's a great point.
65.
Yes.
So sometimes there's age, and then ours, both these trials, the patients have to be on existing therapy. So sometimes they exist. And then one of the key ones that Sarah mentioned was whether or not the patient has existing antibodies to the virus or the vector that it's being used to deliver it. And because these AAV viruses are around us all the time, and there's sort of 12 or 13 naturally occurring versions of these viruses, then humans don't get ill from coming into contact with them, but they do develop antibodies against them because they're a wild virus. And the antibodies that people generate can then make them not eligible for some of these gene therapies. So if you have one of these antibodies, then it might make you not suitable for one gene therapy, but you might be suitable for the other, because these antibodies can be they can work against certain gene therapy viruses, but they may not have a problem with the other one. So, you can only find that out by having the blood tests that are needed for these type of gene therapies.
Yeah.
All right.
I echo all of the answers that Andrew just gave. I also think it's important to know we are interested in reaching younger ages, and because of regulatory requirements, we are sort of mandated to start our trials in adults. So we have had questions about why can't children be included in the trial? Why aren't your ages younger? Health authorities view treating children with a much stronger hand. We cannot go into children if there are adult patients. And so we have to prove that it's safe and it's effective in adults first before we're allowed to lower the age group. So just to add to that, the goal would be to expand on both ends to older and younger patients who are more vulnerable populations in the future.
Yeah, and sorry, just the last bit, I guess people tend not to ask, but most clinical trials won't allow inclusion of, for example, pregnant women or women who may be breastfeeding and things. So there are often restrictions regarding that as well.
All right. Okay. Thanks for that information. So the second question is, how long are the trials expected to last, and what would be the expected cost of the therapy once it is approved?
Oh, that's difficult. So I'll take the first part of the question. So the trials, at least at this time, are required to be 5 years in length. So that's a total follow-up time of 5 years from time of dosing. I would say the first 12 months are typically, the first month is frequent visits. Then we typically move every 3 months. And then after the first year, you move to every 6 months. But the total observation time is 5 years. Some countries actually do require longer length of follow-up. That's typically in something we call a registry, where we're able to follow from a distance or check in once a year. But the trials, because of the newness, the novelty of the therapy, and making sure that it is lasting, that it is durable, that we're tracking any unknown safety events, we have to watch you for the first 5 years. To briefly touch on the question of cost, importantly, as the medical directors of the study, we don't have any influence in the cost. We stay completely separate to the focus on the science and the safety and the efficacy of the study. I think there are lots of information in the news that these types of gene therapies will be quite expensive. And so I think we're watching other therapies that are being approved. For those that may know, there was a different gene therapy approved for a spinal muscular atrophy, SMA, a very different pediatric neuromuscular disorder several years ago. It's called Zolgensma. So people benchmark off of certain other drugs, but because our trial is just starting, I don't want to forecast how much it would cost. I don't know how to predict that, how the financials in the world will look in 5 years.
No, I'd echo that, and just to reiterate with the long-term follow-up, as Sarah says, once you get out, certainly beyond 12 months and even beyond like 2 years, the burden to the patient is much lighter. Often you may only need to be seen once or twice a year for those longer-term periods, and those are mainly, as Sarah said, just to make sure that we evaluate whether it's continuing to work and whether the safety is okay. And some of that is at the request of the global regulatory authorities, who want to be certain that they keep an eye on and keep a monitoring of the safety of these novel type therapies. And the cost is very difficult. And I think Sarah's covered all of that. The only one thing to say is that it tends to be region specific as well. So as well as being disease specific and what available therapies are out there, for diseases where there's nothing else and it's a very high cause of death in children, that may be a very different cost to something where there is other therapies. And each country will have a kind of way of evaluating cost effectiveness and pricing. So it is very, very difficult to say what
I
these
understand.
type of therapies will be on a case-by-case basis. And the world will change a lot, I'd have thought, in the next 5 or 10 years. Yeah.
Right. Yeah. All right. So the next question is, are there any known risks or side effects that are involved in the clinical trials? And are the patients with type 3 eligible to participate?
Sure, I can start with the first one, I suppose. So in our trial, patients with type 3 are not eligible, but I think Sarah mentioned in theirs they may be if they have peripheral manifestations, but Sarah can comment in a second. With regard to potential side effects, as with all sort of investigational or clinical trials or new developing medications, there are always some risks. The known potential risks that people talk about with gene therapies are mainly associated with the way the body reacts or the way the immune system might react to receiving the virus or the gene therapy. And of those, from our point of view, and in most AAV gene therapies, the main risk is around the liver becoming inflamed or irritated from the virus. And we monitor that. And that's why we do such close monitoring for the first few weeks to few months, where we look at these blood tests that tell us how the liver is reacting, because sometimes it can become inflamed and upset. Generally, that can be controlled or prevented using medication like we've discussed, like steroids and other similar medication to prevent that. But there have been reported cases in other gene therapies that have been serious sort of liver inflammation. There appears to be a relationship, though, between the type of virus that's used, but mainly also how much is given. And when you get to much higher doses of virus, then the risk seems to go up. So certainly in our trial, and I think probably the same in Sarah's trial, at these low and medium doses where we're giving relatively low doses of virus, then the risks of liver inflammation or other side effects are relatively low. Other than that, there aren't any other real definitive side effects, and because both of our therapies don't integrate, so they don't go into your natural own DNA, then the risk of that causing a problem in the body's DNA doesn't exist. Ours sit outside, and so that tends to be a different risk from a different type of gene therapy. I don't know if Sarah wants to add anything to that.
No, you've covered a lot of territory. Just to bring it back to the first part of the question, having a diagnosis of type 3 is not exclusionary for our trial. And I hesitate to give sort of personal medical advice here, but if you have type 3 but have mild neurological symptoms, but you also have enlarged liver, spleen, anemia, you could potentially be a candidate. So I think that's really important to discuss with your doctor and with the investigator at the site, when we can look at your specific type of symptoms and decide if it would be a good fit. One of the questions we have is if delivering this in a vein, how many areas it's able to penetrate. We know that ERT may not be good at reaching the blood-brain barrier, reaching the lung, reaching the bone. We have some evidence in animals that is potentially more successful. And so that's another point of this study, is to see where can our gene therapy go that ERT is not good at going? Where can it reach? What tissues can it help that haven't been helped by standard of care to date? So I think those sort of go hand in glove. The questions is we want to include patients that we know would benefit from this IV systemic peripheral administration while we evaluate other places the gene therapy can go. And you covered the safety risks brilliantly. I would just say there are some reported cases of pediatric death in very different diseases. I think it's important to consider the underlying medical condition that those trials were treating. Some of those diseases are very, very different than Gaucher disease type 1. So we have to be careful to learn from those, but not assume the same across populations. In general, our Gaucher community is much, much healthier. much stronger than the patients in that study. So I think we learn from those events. We make our trials safer. For that reason, maybe we draw more blood and we watch you more carefully and we ask you to stay close to the center, close for a few weeks. And the reason we do that is because we want to really make sure you're safe. So we're learning from those experiences, but don't want to assume the same.
Yeah. So we actually had one question which is very related to what you just said about the blood-brain barrier. So one of the attendees is asking if the gene therapy can break the blood-brain barrier.
Yeah, so that is a fabulous question, and I would say that the only way to know that definitively in people is something we're not allowed to do, which is get into the brain. It's something we cannot take a brain biopsy to prove that point. So I think it's something we know happens in mice models. We know that happens in monkey and non-human primate and monkey animal studies. So we are able to see that AAV9 can cross in certain quantities. And that is an open question for us in this clinical trial. There is a hope that can happen in an advantageous way. The other two trials I mentioned that we're running for Parkinson's and Gaucher disease type 2 actually has direct delivery into the, not into the brain, but into the brain fluid called cerebrospinal fluid. There's a direct injection into the cavity in the back to enter the blood-brain barrier, to cross. But the question remains, if we give it in the vein, can it break through the blood-brain barrier that way? So best I can answer is that we've seen it in animals. We're interested to see if it is the same thing we see in people.
All right. Andrew, is there anything you would like to add?
No, I mean, for ours, for Freeline therapy, we don't believe it will get into the blood-brain barrier. I mean, whether the virus gets into the central nervous system, I think is possible. The one thing we've not really gone into in this, because it's, I suppose, quite an in-depth scientific aspect, is that the gene that we have inside the virus has a kind of extra switch as part of it that in our product can only be switched on to work in liver cells. So even if the virus, for example, got into a muscle cell, it wouldn't be able to read the gene to produce the enzyme in that muscle cell. It would only work in a liver cell. And that's called a liver promoter, right? So even if our virus therefore got into the brain cells wouldn't be able to use the gene to make the enzyme. So we think ours is restricted to the liver, and therefore the virus won't produce the enzyme in the brain, even if it got across the barrier. With regard to the enzyme that's released from the liver cells into the bloodstream, we think that's probably going to behave very similar to ERT and not cross the blood-brain barrier. And that's primarily why we have focused on Gaucher disease type 1 for our patients at this time. We are looking at other ways to treat brain-related disease, for example, either in this type of condition or in a similar related condition, Parkinson's disease, that's associated with mutations in this gene as well. But we're not, that's very early. You know, we don't have that in this current trial.
All right. And we have one question, which is probably the last one. So what effect would the gene therapy have on aspects of Gaucher, like lumbar vertebrae fractures or bone lesions?
It's a really important question. So I'll just say that's another slight difference between our products, is that the switch we have is not specific to the liver. It's a more general, open, the science word is ubiquitous. So sort of not cell type dependent. It can go many places and be turned on several areas. So much like the blood-brain barrier, the bone cavity and the bone marrow, we know that's an area of much pain, burden, Gaucher cell accumulation, and especially as patients age, that can be really a difficult part of the therapy that ERT can't help. So that is for the best we do is our animal studies. We do evaluation to see where the virus is able to reach, what cell types it's able to reach. Is it able to reach the brain? Can it reach the bone marrow? And with that, we go into human trials with certain doses with confidence that if we're able to see it in mice and in monkeys, are we able to see the same thing in humans? So I would say the lung, the brain, and the bone are the three cavities that we're most curious and hopeful about would have an effect above and beyond ERT. Unfortunately, it's the point of the study. I don't have an answer. The hope is yes, but I don't have a good answer for you. We've only dosed one patient. That is a hope. That is a strong hope, and we recognize it's an unmet need in the community. We recognize it's a big source of symptomatic trouble. So I think whatever therapy we're developing, our hope is that we're able to make enough enzyme, or we're able to reach those tissues in a way that's helpful.
Andrew?
Yeah, and very similar to ours in that Freeline recognize that certainly the bone and the deep tissues are areas that may not have been fully addressed by ERT or SRT, and are similar to Sarah. That's the purpose of the trial. We don't know yet really what the effects are in the bone. We need time, to see effects on things like lumbar vertebrae fractures or the effects on bone thinning. You need quite a long time, months, to see whether it's having an effect in more than one person, right? So we'll collect those in the trial. Our data from the animal studies suggests that the enzyme that we make, we get good levels of that enzyme in the bone and in the lung, and it's able to remove the Gaucher harmful substrate and the Gaucher cells from those bone and lung regions. So we're hopeful it will work in humans, but we don't know yet. So the vision, I'm sure it's the same for Sarah and Prevail's therapy as ours, is that these therapies will hopefully be able to reduce the likelihood of bone fractures and bone fragility and improve bone strength, but we just don't know yet.
All right, thank you. So there seem to be no further questions. So I think that brings us to the end of this webinar. And I would once again like to thank both Sarah and Andrew for sharing this very important information with all of us. And I'm sure the audience has learned a lot, and it will encourage them to consider participating in these trials or even spread the message to others who might be eligible. So if the audience has any more questions, you can email us to them, and we'll do our best possible to have them addressed after the webinar concludes. So once again, thank you, everyone. Thank you to all the speakers for having, sparing their time with us, and all the attendees as well for their active participation. So thank you and have a good day.
Thank you for moderating, and thanks to the translators. Thank you.
Yeah, thank you to everybody and to IGA and all the participants. Thank you very much. Nice to meet everyone.
Thank you.
Goodbye.